Hello everyone, welcome to the Bodhi Academy. In today's video, we'll learn about paper chromatography. So, paper chromatography is a technique which is used to separate different molecules or different pigments so that we can observe and identify those. Okay, so first we have to do is we have to take a filter paper and cut it in this shape. Then with a scale we have to mark a line about 1 centimeter above this point. Okay, now we have prepared a leaf slurry, that is we have grinded spinach leaves with acetone, okay, 5 ml acetone. And now we will take this capillary tube, take the slurry in the tube. See, you can see the green color slurry, and then try to load it at the center of this line. Now we have to let this loading dry, and then again we have to take and load it for 4 to 5 times and then let it dry. Okay, so now our chromatography strip is ready. Then next what we have to do is we have to take a chromatography chamber, and in that we have to pour a solvent. Okay, for example we can add ether acetone in this. Then what we have to do is now this is our chromatography chamber, and in this the solvent is present. Then we have to take the filter paper and try to suspend it just as the tip touches the liquid. Okay, so now what will happen, the solvent will slowly rise on the paper strip, and just when the strip or the solvent has reached about a 3/4, at that time we have to remove this filter paper and then let it dry. Okay, so now as you can see here, as the solvent was rising, the different pigments of the leaf that were present in this mixture or the loading that we have done, they started separating. Okay, so now here you can see the different pigments of leaves that have separated. So the first one that we have, this yellowish green color, is chlorophyll B. Then the dark green color which we have is chlorophyll A. Then the yellow color pigment over here is xanthophyll. And then the topmost one, that is orange yellow kind of, that is the carotene. Okay, now this is the line till where the solvent has reached, and it is called as solvent front. And this is the origin where we had loaded. So let us connect these dots and see the height or the length till where these pigment have reached. So the solvent front is 7 centimeter. That means the from this start point the solvent has travelled up to 7 centimeter. Then the next one is carotene, which is 6.73 centimeter. Then xanthophyll has 5.26 centimeter. Chlorophyll A has reached 4.46 centimeter, and then chlorophyll B is 3.03 centimeter. Now why we have to measure these lengths? Now this is because using these lengths we can calculate the RF value of each of these pigment. Okay, so RF value is equal to the distance travelled by the pigment divided by the distance travelled by the solvent. Okay, divided by the solvent front. So let us see. The first one is the yellow orange. We have to make a observation table which will include the color, RF value and the pigment. So the first one was yellow orange, and it travelled up to 6.73 divided by the solvent front. So the answer is 0.96, and the pigment was carotene. Similarly, we have the RF values of xanthophyll, that is 0.75. The pigment, the color was yellow. Then we have the chlorophyll A, that is 0.63. The color was dark green. Then chlorophyll B, the RF value is 0.43, and the color was yellowish green. So this is how this chromatography technique is used to separate different pigments or different molecules in a mixture. Now this is all in this video. Thank you for watching my video. Hope you liked it. Please like, share and subscribe to the Bodhi Academy.